All right, Chapter 6, Cellular Respiration. This is the other side of the photosynthesis. Basically, the products of photosynthesis are used by cellular respiration in animals. You're going to want to pay close attention. This one is a little involved. Okay. It's basically its definition. It's a cellular process. It requires oxygen and gives off carbon dioxide. Usually involves the breakdown of glucose to carbon dioxide and water. The energy extracted is released stepwise. It allows ATP to be produced efficiently. The oxidation reduction enzymes include NAD and FAD as coenzymes. But let's look at the very beginning. Respiration basically is when you take glucose, and you want to memorize that molecule, C6H12O6, plus 6 oxygen, and that's what we feed into cellular respiration. And then our waste products are carbon dioxide and water. Now, this is called a redox reaction, all right? And the reason it is called a redox reaction is it takes these hydrogens, which have high energy electrons on them, and transfers these hydrogens after it bleeds off the energy from these electrons to water. Okay, so when this glucose, gets rid of its hydrogens, it becomes carbon dioxide. And then the oxygen picks up the hydrogens with the high energy electrons and becomes water. And then you get energy, and the energy we get here is in the form of ATP, adenosine triphosphate. All right. So glucose is oxidized because it loses the hydrogens with the high energy electrons, and the oxygens are reduced to water because they gain the hydrogens with the high energy electrons. Now, why do they call it reduction? Because if you remember, electrons have a -1 charge, and so when this picks up these high energy electrons that are attached to the hydrogen molecules, they decided to call it being reduced. Okay? This is a mitochondria. All right, and inside the mitochondria are these little membranes called crista. Okay, and then inside here is matrices. These are organelles, if you remember from chapter 3. And these organelles, basically their job is to produce ATP. Now, interestingly enough, mitochondria have their own DNA. It's unique from the DNA of the host. This gives rise to the hypothesis, and there's a lot of evidence to support that at one point in an early cell, a bacterium formed a symbiotic relationship with an early cell and was included. And so we can actually follow lines of DNA in these mitochondrial DNAs. Why do we think it was once a bacteria? Well, it has its own DNA. It doesn't have a nucleus. It just has a nucleoid area, just like a bacterium. It's got a cell wall, a cell membrane, and it's basically built like a little miniature bacterium. Okay, so whether we'll ever know 100%, probably not, but I think there's enough evidence to really support the hypothesis that at some point this was its own bacteria. NAD and FAD. A lot of people have trouble understanding these. NADs and FADs are coenzymes that just pick up hydrogens with electrons on them and carry them from one spot to another. That's it. It's that simple. All right? They are just a transport molecule. They don't do anything else. Now, in your lab, in your lecture, PowerPoints, these little blue buttons usually have linked to videos like this, and you can just get to it and go open hyperlink. Some of you, depending on your computer and your settings, might be able to play them if you start a slideshow from the current slide and then just play it.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these
Now, I can't play this whole thing or else they will remove my video for copyright infringement, but I can direct you to where they go here. Basically, I will do some reviews over these of how good I think they are, and you can use parts of copyrighted materials for a review. So I will be doing reviews of these videos to kind of give you an idea. But NAD literally, and FAD is just another similar molecule, and why it was built, it's just random chance, but they both do the same thing. Just carry a hydrogen is what they do. So these are the 4 phases. Glycolysis. Now, some people don't consider transition reaction a phase. Okay. So be it. But we're going to look at it as a phase. We have the glycolysis, the transition reaction. We have the citric acid cycle. The citric acid cycle is also called the electron transport system. I'm sorry, no, that's this one. Citric acid cycle is also called the Krebs cycle. Sorry, it's early. I'm trying to get some of this done before my lab starts. Glycolysis occurs outside the mitochondria in the cytoplasm of the cell. All right? And the prep reaction takes the product, pyruvate, from glycolysis and converts it into acetyl coenzyme A. And so that's done at the membrane of the mitochondria, the entrance, before it gets in. And then the Krebs cycle and the ETS occurs inside the mitochondria. So let's try to find how this works. If I can find a... Here, let's look at this. So, how does glycolysis work? It's really quite simple. What you have, you start with your glucose, right? C6H12O6. And the first thing that happens to the glucose is you spend 2 ATP to start the ball rolling. ATP are energy molecules. So you spend 2, and it will split this into 2 molecules that are phosphated. All right? And then they split. And NADs pick up some hydrogens, and they make some ATP. Okay? So what you've got, you start out, you spend 2 ATPs. Okay? Go over it once again. So I spent 2 ATP. And remember, when you see a little line, that just means a series of chemical reactions. So we have chemical reactions going on here, and we're spending 2 ATP, and when we spend them, we now have 2 ADP, 2 adenosine diphosphates, and we take that third phosphate from both of these molecules and we attach it to the glucose. When you attach an energy phosphate from an ATP to a molecule, that is called phosphorylating the molecule. Very, very important to remember that. Okay? So you phosphorylate that glucose. Now, we go through some more reactions, and we split this in 2. So now we have 2 molecules. And they continue going through reactions. And the ones I want you to remember here is we get some NAD and we convert it to NADH. This NADH is going to be carried to the electron transport system, where the electrons are going to be separated from the hydrogens and the energy bled off to make ATP. But through substrate level phosphorylation, we are going to make 4 ATP, 2 on this side and 2 on this side. And remember, we spent 2 up at the top, so it cost us 2, we made 4, which means we had a profit or net gain of ATP from glycolysis of 2. Okay? And at the very end, we get this pyruvate. Okay? And the pyruvate is going to be converted in the transition action or transition reaction to acetyl coenzyme A. So glucose to pyruvate. And you'll see these NADHs we made, they're going to go all the way down here to the electron transport chain. It's in the hang on, I'm trying to get this to work. Not sure what you're seeing here, so let me end the show. There. Now you should be able to see it. Yeah, the glycolysis. The glucose goes to pyruvate. Those NADHs we made there goes down here to the electron transport chain. Now, pyruvate has to undergo transition, but there is a way that there's 2 ways that things can happen in glycolysis, and one that we need to mention. Now, as long as there's oxygen, glycolysis functions just like I told you. And it produces this pyruvate, which is also called pyruvic acid. If you run out of oxygen, then what happens is fermentation. Fermentation is anaerobic. Anaerobic, might want to write this down, means without oxygen. And during fermentation, we can do the same thing, okay? But instead of getting pyruvic acid, we get lactic acid, okay? And lactic acid can be detoxified and washed away. But to give you an idea of how this works, let me use this example. If you've ever went to the gym, okay, and you start working your muscles, eventually your muscles will start burning, okay? And what that means is when you run out of oxygen, but you're still using your muscles, the only way they can make some more ATP to keep lifting is to start going into fermentation, anaerobics. We run out of oxygen in our muscles. And we'll talk about this, well, if you take 1020, we'll talk about muscles and bones and how that works. But feel the burn means you basically exhaust the oxygen in your muscles, and so they're still trying to produce ATP to keep contracting the muscles, and so they use fermentation, and that lactic acid gets into your muscles and causes a burn. Literally, it's an acid, and it causes some pain. Now, if you keep going every other day and you keep working out, eventually you might feel that little burn, but the next day you're not all sore, and that's because your muscles will adapt. If you start using your muscles more, they'll start putting more blood vessels growing into them, bringing more oxygen and more ATP. All right? And they'll also respond by getting stronger and getting bigger. And so if you've done weight training, once you get to that place where you're not feeling any pain anymore, then you got to up the amount of weights you're lifting. And then you'll start getting that burn. But once you've started the process, when you move your weights up, you won't be that sore the next day unless you move them up too far. Okay? And your system is more efficient at that point in time. It is actually getting out the blood there, and it's washing away the lactic acid very quickly. The lactic acid gets detoxified in the liver, okay, which is an important aspect of that. Now, the end product of glycolysis is pyruvate. It enters the matrix, and it gets converted into 2-carbon acetyl group, which we call acetyl coenzyme A. Now, in the coenzyme A, the acetyl coenzyme A, in that process of being converted from pyruvate to acetyl coenzyme A, you will sometimes generate some NADH, okay? And you'll release a little carbon dioxide, which you'll breathe out through your respiratory system, all right? Now, I want to touch base here. Substrate level phosphorylation. Remember I mentioned up here when we were looking at how glycolysis worked, we made the ATP using substrate level phosphorylation. This is what that is. The enzyme is a protein, okay, and it has a slot that holds an ADP and a slot that holds a phosphate. And what it literally does is this part folds in and this part folds in, and it presses this phosphate from this molecule to this molecule to make ATP. It is not the most efficient way to make ATP, but it works. Okay? And it doesn't make as much ATP as the electron transport system does, which is oxidative phosphorylation, but we'll get to that shortly. Now let's go back to the preparatory reaction. The preparatory reaction is when we convert the pyruvate to acetyl coenzyme A, get some NADH. The NADH from the prep reaction goes to the electron transport system, okay? And what happens, the NADH, it goes down here and it gets rid of the H's, and then it comes back and gets more H's. That's all it does. It just picks up an H in one of these phases and carries it to here. And that's all it is. It's no more complex than that. All right? Just picks it up. Here is the prep reaction. We make some NADH. We go from this molecule to this molecule. And this molecule becomes the fuel for the next phase, which is the Krebs cycle. And I'm about out of time this morning, so I'm going to go ahead and put part 1, glycolysis, out and get that going. And then today or tomorrow, I will try to do the other parts. Okay, so we've covered the glycolysis and we've covered the prep reaction or transitionary reaction.